everybody, and I hope you're having fun with what we're presenting today. It is great to be back to share a little bit of what we're doing in our multiomics portfolio, how we're using the power of the X and of all our sequencing platforms to continue to give you more reach and more abilities. We know that biology is complex, and the questions that we want to ask are also complex, but we were limited by tools historically, either because the tools were expensive, they were disconnected, or unscalable. That forced people to look one layer of biology at a time. And that meant that science was restricted by the tools that were available and not by the questions or our ability to ask those. So last year at this same conference, in a different stage with a little better weather, I had the privilege of introducing to you some of the advancements and tools that we were bringing in for multiomics. These tools were meant to unlock your capabilities and give you new layers of biological insights across the genome, the epigenome, the transcriptome, and the proteome. All of this powered by AI analysis to give you more integrated insights. We are committed to providing full workflows that allow you to have a more complete view of biology. We want you to spend your time on the things that matter, not on the things that are giving you heartache. So today, I'm gonna share some updates on what we're building in this suite of powerful multiomics and multimode workflows. When I talked to you about this a year ago, we had just launched our Illumina single-cell kit. Illumina protein and 5-base and connected multiomics were either in R&D or in early access. At the time, I said that we will deliver on these capabilities in 2025, and I am very proud to say that we did exactly that. These workflows are launched, they're in customer hands, and people are producing data that is remarkable. You're going to hear some of it later today from our speakers, and you're going to see that in many of the posters and talks during this conference. Looking ahead to 2026, I'm very excited about what our Illumina Spatial technology is doing. The feedback that we're getting from customers is great, and we're still on track to launch mid-year. And this year, something pretty cool happened to us. We acquired SomaLogic, like many of you may know, a leader in proteomics technology. And that was a very intentional move. It completes our multiomics strategy in a way that we are able to provide scalable, high-quality proteomics insights. It's about expanding what is possible, not just adding another modality. By combining the expertise and the technology, our two teams are able to accelerate innovation and expand access. Somamer technology actually delivers precision, scalability, reproducibility, and specificity in a way that no other tool or method could do before. But it's not just about the technology. The other thing that happened is we have an amazing team with 20 years of experience in proteomics. Together, that allows us to continue evolving this technology to reduce the time and cost. Look, let me show you a little bit of what we did already together when these two teams came and executed. Last year, we launched the most powerful NGS-based protein solution in the market. Twice as many protein targets as any of the competitors out there, representing more than 50% of the circulating native proteome. This gives users power and scalability with the highest precision and very high sensitivity. That means that the studies can be large and you're only paying half the cost per insight than any of the other solutions out there. The product launched last year. It's already in 20 sites installed, growing its install base, and I'm happy to share that soon we will have several public datasets that you can access to reference to your own datasets and make your studies more powerful. But this is only the beginning. Now, by being one company, we're going to be scaling proteomics with the Somamer technology beyond the proteome. We're going to bring it into functional proteomics. We're going to bring it into spatial proteomics as well. In our roadmap, in the short term, we want to continue to unlock those capabilities of this technology by enabling larger and smaller panels. We have a clear path to the full native proteome in our roadmap. We're also going to be enabling new sample types, and all of this will happen before the end of this year. Together, we want to bring protein-based disease risk prediction tools that will come later in 2027. And in our R&D roadmap is full and rich, and we're going to be bringing the technology to expand it across modalities. We want to bring aptamers into proteomics and multiomics. We want to expand beyond native proteome into functional proteomics and also look for other analytes. And at the same time, we want to make sure that we continue to improve on the workflow, simplify it, and continue to lower the cost. So we don't want the tools or the cost of the tools to be what limits what you can do with them. Another great example of how we're actually bringing this ecosystem together is our 5-base solution. It's you put one sample, one library prep, and one analysis that provides dual insights. Genomic and epigenomic variants are being detected at the same time. And you may wonder why that matters. It's because by having those two things together, we're able to better classify cancers in a way more streamlined way than any of the other solutions out there. And you're going to see that from some of our speakers later today. This product launched late last year and is already having widespread adoption. Users are really enjoying what they're getting out of it. It's been used in cancer, rare disease, and population health. It is powered by Dragon pipelines for somatic and germline pipelines, and it supports a very, very broad range of applications, as you can see on the slide over there. So what we're enabling when we launch our single-cell kit, so changing a little bit into the other pillar of our multiomic strategy, was shift and scale and economics. We brought the cost of single cell down so that now very large studies, between millions and billions of cells, are possible. With the Proteomics kits that we have in early access right now, we're able to build a billion-cell atlas across thousands of perturbations per cell line, hundreds of diverse cell lines, and primary cell cultures using the same PIP-seq technology. We're partnering with pharma and biotech and consortia to get this atlas available, and it will be available to reference. But that doesn't stop there, because we don't want a few players to have access to that data. We want everybody to do it. We want to democratize access to this type of large-scale studies. So we are launching our Proteomics distributed kit. So labs of any size, without needing massive infrastructure, can do the same kind of studies. We're very proud to continue to expand this. Now I'm going to tell you a little bit about our last pillar, our spatial product. It is showing remarkable results on the field, in the wild. It's released in early access. We've engaged with more than customer sites, with projects ranging from oncology, neurobiology, plant biology, and others. And when we launched this product, we proposed a different value proposition. It was large area, whole transcriptome, 4 times lower cost per unit area than anything out there, and 4 times higher sensitivity. What we thought was this was not going to be about incremental gains. It had to be about seeing biology that was previously invisible. Here on this video on the right is a 3D reconstruction from a pig-to human kidney xenotransplant biopsy that Brendan Keaton and his team at NYU did. They were able to see thousands of cells in their native context and seeing patterns that were not possible to see with any other technology. By lowering the barrier of of discovery, we're able to enable studies that are much larger and impossible before. And we will continue to do so. We're committed to enable for you to do more with what you have. We're on track to deliver this product later this year, so I'm excited to see it in the hands of everybody. And the R&D roadmap is also very full with enabling FFPE, enrichment panels, and proteomics. But that's not all. None of this matters without analysis. And I'm a chemist by training, so when I think about people having to do analysis, I was dreaming of the age where you didn't need to know how to write lines of code, and that is here. Illumina Connected Multiomics brings interactive visualizations, powerful algorithms, customizable workflows, but also default workflows, and scalable analysis. So all of us, all researchers, can move from actually generating data to actually understanding biological meaning. It works for small labs and large consortia without having to change tools. So that allows a lot of flexibility and brings the power of all of these assays and sequencing and pipelines for analysis to the hands of everybody. We are committed to continue to invest in these tools. And you can see here the release plans that we have for 2026. We want to continue to expand that AI-assisted analysis. And my favorite one is the chat-based conversational support. So you can just ask it questions, and it will help you look for those rare cell types, those rare transcripts in Spatial, for example. It will bring multiomic integration, and you're going to see some of that in some of the talks later today, and improving usability. So for us, the goal is super simple. We want you to be able to ask better questions and get your answers faster. So for me, the key takeaway is this. That insight ecosystem that we've been building is here. It's available to everyone. You can now generate deeper insights without compromising on throughput, cost, or power across a variety of omes, across a variety of sample types. It's a more complete view of biology with the highest quality insights at the lowest end-to-end cost. You're going to see in the talks that follow me how 5-base improves cell-free DNA resolution, how proteomics strengthens interpretation, how we can use spatial biology to disambiguate confusing answers, and most importantly, how ICM brings it all together. The insight ecosystem is here, it's available to everyone. And I am very happy to show you how some of our customers are using it today. So thank you so much.